excited to talk about my work, recent work looking at DNA methylation and insects and its role in social behavior. So for those of you who don't know anything about DNA methylation, a little quick crash course. It's chromosome modification. It's the addition of a methyl group onto the 5' carbon of cytosine. And broadly, this modification protects the genome from altered genetic elements like TEs or other repeats. Sometimes important for proper regulation of gene expression, is involved in the stability of the genome, so distinguishing heterochromatin from euchromatin. And in animals, specifically, this type of modification is more often not found at CpG sites, so cytosine followed by guanine. And when it's methylated, the site is methylated, we refer to it as an MCG site, just for technical purposes. And in insects, specifically, this type of modification is enriched in coding regions. So lots of DNA methylation within coding regions in insects. At the population level, there exists quite a lot of epigenetic variation, and researchers like myself and others are interested in how this type of variation can contribute to complex traits. And so there's been some nice work done in Arabidopsis thaliana looking at this. However, outside of model plant systems like Arabidopsis that can be easily manipulated, where we can do things like epi-QTL mapping and create epigenetic recombinant inbred lines. The majority of other organisms sort of go unnoticed, and we don't really know anything about DNA methylation and how it might contribute to other complex traits. But we're not giving up, so really we can turn to other organisms and use comparative phylogenetic methods to test hypotheses on the relationship of DNA methylation to other traits. So really what we're going to do is we're going to have DNA methylation as a trait correlated to other traits out there. So in particular, the be-all and end-all of DNA methylation in insects is social behavior. So it's somehow involved in regulating different behaviors of individuals within a single population. That's all the way from the evolution of social behavior down to caste differentiation, so it's very extreme eusocial behavior. But the majority of studies out there in literature are really restricted to pairwise species comparisons within the order Hymenoptera. So this includes your ant species moths. But we know that sociality in insects evolved many times, and then we also know that DNA methylation can be found in solitary insects like the silkworm. So with this sort of framework in mind, we took a much broader phylogenetic approach. And we decided to look at DNA methylation and social behavior across the insect life. So every single order we sampled from, except for Orthoptera, which are your crickets and grasshoppers. Again, we did phylogenetic comparative tests, and we did two different types. The first one is correlated evolution of two binary traits. This was described in a Bewick paper in 1994. So we have our two traits again, DNA methylation in binary, so presence or absence. From 123 species from 11 different orders of insects. We have our social behavior, again, coded in binary, social or solitary, or not they have castes or don't have castes. So these 123 species don't have a methylome or any whole genome bisulfite data available per se, but we can predict whether or not it has DNA methylation based on this predictor, the CpG-OE ratio. I'm sure many of you are familiar with that, but if you're not, here's a little quick summary of it. It's the proportion of CG sites normalized by the number of cytosines and guanines at a particular locus. Could be a gene, could be a whole chromosome, could be a window within the genome. And this predictor hinges on the theory that methylated cytosines deaminate over time. So you'll have a methylated cytosine guanine pair that will deaminate to a TG pair. And so then if we have a mixture of genes within a genome, hypermethylated loci then should have a low CpG value, CpG-OE value, and then hypomethylated loci should have a higher CpG-OE value. So this is sort of proof of concept. We have honeybee, fruit fly, and German cockroach, 3 different insects. We know from whole genome bisulfite sequencing that honeybee has DNA methylation and fruit fly doesn't, and this is reflected in their distribution of CpG-OE across all genes within their assembled genomes. So honeybee has this nice bimodal distribution where you have hypermethylated loci and hypomethylated loci. Fruit fly does not have this type of transition or deamination going on, so it has this unimodal distribution. And for German cockroach, given this distribution, we would expect it to have DNA methylation. I'll show you later on, using whole genome bisulfite sequencing, that it actually does. So we did this for 123 species from 11 different orders. We ran Pegel's tests, and what did we find? We found that there was no support for dependence between social behavior and DNA methylation across all insects. So here's the tree that we controlled for phylogeny for. Here's our traits coded in binary in these black and gray boxes for presence or absence. And over here I have a collapsed tree down to the order level, again showing you this type of variation of presence absence and how it doesn't necessarily line up with social behavior for DNA. Or DNA methylation doesn't line up with social behavior all the time. Again, the previous study is really focused on the order Hymenoptera. If we were just to look at Hymenoptera, you might make this assumption that, yeah, there is this like pretty strong correlation between DNA methylation and social behavior, but it's not until we take this much broader taxonomic approach that really this trend sort of breaks down. The second test we performed is a PGLS test, phylogenetic generalization of squares. Slightly different test, slightly different data going into it, where we can have DNA methylation coded as a continuous trait. So we did whole genome bisulfite sequencing from 40 different insect species, 32 of which we generated novel data for, so 32 new species of insects from 6 different orders. We had a little more liberty in how we can call our other traits, so our social traits. So we categorized insects whether or not they were eusocial, subsocial, presocial, or solitary. Fully admit this is a relatively small number of species, but I think it's a very important number of species because they represent orders for which we know eusociality has evolved independently. I have starred here, so you get your Blattidia, which include your cockroaches and termites, your hemiprins, which are your true bugs, and then your Hymenoptera, so your anti-human wasps. So what did we find? Again, after we ran this test, we find no correlation between social behavior and DNA methylation, which is interesting. It goes against what has been published previously. So here's the tree that we use to control for phylogeny. Here's our social traits coded in these colored boxes. So again, the 4 different categories we can pin them as. Here's some 2 bar plots showing cgDNA methylation within the genome and at coding regions for all of these 40 different insects. And I have here boxed in these different orders that have independently evolved eusociality. And what you can see is that there is a substantial amount of DNA methylation variation across all these insects. And that the highest actually can be found in the order Blattidia. And there's other little things you can do apart from this. For example, these 2 eusocial termites have very different levels of DNA methylation despite being eusocial. And down here we have these 3 different Comparative aphid species that have very different social behaviors but have pretty similar levels of DNA methylation. We pursued some of these interesting observations a little further. The one that we jumped on right away was this outlier, because outliers are always cool to look at. So we performed some deep whole genome bisulfite sequencing, and guess who was the highest? This solitary German cockroach species that I showed you before, this distribution of CpGOE. So here I'm showing you 2 metaplots looking at DNA methylation upstream of genes, within genes, and then downstream of genes. For 6 different species, I got 4 insects here, Arabidopsis and the pufferfish. So over here, the y-axis goes from 0 to 4% methylation, and this includes, in this plot here, the species are Bomix mori, so Necrophis aspeloides, the bearing beetle, and Apis mellifera, the honeybee. Over here, though, the y-axis goes from 0 to 80% methylation, so this plot here is really squished down. And you can see that the German cockroach lies nicely between Arabidopsis and the pufferfish, and qualitatively, that the distribution of DNA methylation across these regions in cockroach are more similar to that of a vertebrate than they are to an invertebrate. So we're pursuing why that might be and other sort of genomic nuances of the cockroach genome and whether or not it's more similar to a vertebrate than invertebrate. So in conclusion, we find no evidence for relationship between sociality and DNA methylation. So really I think we need to start rethinking the role of DNA methylation in insects, and just sort of a back of the envelope alternative is that maybe social behavior is mediated by other factors like the environment or hormones. And then methylation then is a downstream consequence. We also find that there is variation in DNA methylation in insects, and it's somewhat analogous to what we see in plants. There's lots of variation. Lots of variation we actually don't really understand a whole deal about. And that the highest levels of DNA methylation can be found in solitary insects. So directions and research plan, if I get a job, is to continue with this type of work, looking at evolution of mechanisms and patterns of protein modifications across the tree of life, so really bridging epigenetics and phylogenetics together. And then once I have this nice framework, we can begin to test hypotheses on how natural selection might shape function modification. So looking at selection on these different types of enzymes and how that is reflected in the epigenome. I've already built up some strong collaborations with the 1KP, so 1000 plant group, the 1000 fungal group, and the 5000 insect group. Pursuing this pretty heavily. So with that, I'd like to thank many people who've helped with this, from samples to lab work to just discussions of this study, particularly Patrick Abbott and Chloe Shaw for providing aphids and cockroaches that were key to this study. GGF for sequencing resources, GACRC for computational resources, UGA, Pew, and NSF for funding, as well as everyone here at Evolution, from those who organized the event to those attending. Thank you very much. And I'd be happy to take questions. My email's up here. I'm pretty good at answering email. I recently became a father, so I can answer emails at 3 in the morning often when I'm up. So it's also ironic that it's my first Father's Day, and I'm giving a presentation. Hey. I think I whizzed through that talk. Yeah? Yes?
The methylation that causes depletion of CPG islands, is that at the germline, right? Not the, but presumably it's methylation of the soma, that would have effects on social behavior and so on. So is it a good assumption that what you measure with sequencing from mostly somatic DNA is indicative of the state of the germline?
I think there's a lot that we don't know in terms of reprogramming during insect embryogenesis or even development, and so I don't really know how reprogramming is occurring in insects, because there are lots of insects out there that don't have this de novo methyltransferase but have DNA methylation. So I would guess, though, based on the presence of the maintenance of the de novo methyltransferase, that there is reprogramming occurring, so this is likely being reestablished each time. So it's one of the things that gives us so much. Yes.
So for the 40 methylones you did, so was that base resolution or was that the genome-wide methylation? And then for the 6, you clearly had base scale resolution.
So we did, it was just very, it was ranged in the amount of coverage, but it's whole genome by sulfide sequencing.
Okay.
So yeah, so it's weighted methylation, so the number of cytosines over the total number of cytosines that are methylated.
So I know the hypothesis was that CPG methylation was responsible for potentially some of these changes in behaviorality, but did you also look at CHG or CHH context potentially, even though the changes might not be as extreme as presence-absence?
So there's actually been no evidence that other type of context exists in insects. So, I mean, it's something that it's worthwhile to look into, because maybe there is some of that, but I did not look at that context specifically for those contexts.
I have another one. Ah, shit.